Thank you. Our next speaker is Jose Rodriguez, who's my a colleague at UCLA and who is heading up the efforts for the microED and associate professor at UCLA. So we're really glad you could come today, Jose. Thank you very much. Looking forward to your talk.
Wonderful. Thank you. Let me start sharing my slides here. Thanks for the opportunity to tell you a little bit about the opportunities for atomic structure visualization that we're trying to set up at the BioPACIFIC MIP. I do have to say that a lot of the group that I'll be, a lot of the work that I'll be talking about today was done by my group, and I'll acknowledge them at the end, but throughout the talk, I'll basically be highlighting some of the efforts that some of the members of my group have participated in. And I want to start by saying that a lot of the knowledge of structures at the molecular scale and at the atomic scale rely on our ability to interrogate assemblies that molecules form. We see them in our daily lives as crystals, and crystals can be as big as those of a grain of salt or even larger, and you can handle them, grab them. At the atomic scale, they are simple repeats of molecules all ordered in a very particular arrangement. And crystals have allowed us to understand structures of molecules as complicated as insulin. You see that on the slide here. That was determined by Dorothy Hodgkin. Regular ordering has allowed us to understand the structure of DNA from data collected by Rosalind Franklin, and in the earliest days allowed us to understand the structure of matter itself, the fact that atoms are ordered. You can see a crystal of simple sodium chloride here on the left-hand side, whose structure was determined by Bragg Sr. and Jr. Now, we study molecules that assemble into crystals, sometimes big enough for us to see, many times small enough that we can't actually see them with the naked eye. The molecules themselves, of course, are so small that you definitely can't see them. They range from about the size of a molecule of glucose, which you individually cannot see, to as big as a virus, which you still can't see with the naked eye. We also can't see them with a light microscope, the type of microscope that you're used to visualizing or magnifying things with. If you were to take an assembly of these molecules and put them into a light microscope, you might be able to see an image shown on this slide up above. You see a very nice crystal of a molecule here that is self-assembled, and the crystal is basically transparent, so light can go through it, but you see the edges of the crystal, and the edges of the crystal give rise to this geometric shape. So you see a sort of hexagonal shape that reflects the symmetry present in the arrangement of molecules within that crystal. If you were to zoom in to that crystal, you would actually be able to see individual molecules lined up one right next to the other, obeying a very particular set of rules in their packing. And that allows us to then interpret the average structure of a molecule within this crystal. So all of those molecules contribute to signal that we interpret and then understand as sort of the average molecule in the ensemble. That does mean that all of the molecules have to be basically identical, and they have to obey the rules of packing very precisely. Any deviations from that will obscure our interpretation of what's present within that crystal. And those deviations abound. Many times molecules are only able to form crystals of very limited size. And in those cases, those crystals are not only hard to find and manipulate, but they're even hard to see. So here are an example of microcrystals. These crystals are so small that you definitely can't see them with the naked eye, but you can sort of see them under a light microscope. And they're lined up here against the edge of a dime. You can see the E in a dime here. And you can see this is a montage or composite image of many glass capillaries. The glass capillaries you can see pretty readily on the slide. And at the tip of those glass capillaries, sometimes you can see a little needle, a crystal hanging off of the tip there. And those are the crystals that we would interrogate to try to understand what the average structure of a molecule that has made that assembly, what that average structure is. There are even smaller crystals than that. If you turn that dime on its side and you look at 1 pixel of an SEM image now of that dime, here's a surface of the edge of that dime. 1 pixel would be even bigger than one of the nanocrystals that we use to interrogate structures of molecules with. And so these are really small crystals. How do we deal with them conventionally? Well, for many years, the thought was that if your samples are so small, we really just need to supply more energy, more light, to try to interrogate them, because where there wasn't very much material to actually produce signal from that light, you simply just need to pack more light into the interaction and then get more signal out. And we've done that for many years. We've built enormous infrastructure to try and do that. Here are examples of the types of machines that are used to do that. These machines are as big as a building, or even many buildings large. One example is of the Advanced Photon Source in Chicago. These are giant rings, and light is produced at locations along this ring. Electrons are actually accelerated within the ring to produce light or X-rays at particular locations. Each of the locations then has an end station, a whole lab associated with it, to try and interrogate molecules at that location. Here's another example of one of these circular machines. This is a Spring 8 synchrotron in Japan. I've been to this particular synchrotron a few times, and what's nice is that when you're taking a break from your experiment, or getting a little bit weary of sitting down and taking data, you can actually climb to the top of the mountain that's actually in the center of the synchrotron. So the synchrotron is so big that it encircles a mountain, and you can see the mountain here, and you can climb to the top of the mountain. So that's kind of nice, but you can get a scale, a sense of the scale of these machines. They're pretty enormous. Another example of an even more powerful machine is this linear accelerator that's found at Stanford. And in this case, you are packing so much energy, so much light into such a small space that you're doing so very brightly and destroying everything that you are interacting with when you emit this light. And so this light travels down this linear path, eventually reaches an experimental hall that's actually dug out of the side of a hill, and interacts with your sample and destroys it. And this is a 1 kilometer long instrument. So you can realize that if your samples are really small, it's not going to be easy classically to try to interrogate their structure at the atomic scale. Now, I've mentioned 2 types of instruments that are used for this purpose. So synchrotron X-rays and X-ray free electron lasers. What I'm going to tell you about today is an alternative that is at the laboratory scale. And so not going to a whole building that encircles a mountain, or a 1 kilometer long instrument that is dug into the side of a hill, but an electron microscope that sits in a room much like the size of your living room. And that's been around for many years, and that is available in many universities. So an example of this instrument now for scale is shown here. So this is a transmission electron microscope. It's hidden inside this box, so you actually can't see the contents inside the box. But you can see Dr. Marcus Gallagher-Jones, who worked in my group for several years and is now in the UK pursuing electron microscopy work. You can see he would climb these stairs and put a sample into this instrument, and the instrument is about maybe twice his size. Within the instrument, an electron source would illuminate ordered array of molecules, demonstrated here as yellow spheres. So you can see a crystal here of yellow spheres. You can imagine that those yellow spheres would indeed be molecules themselves. And from that interaction, you would try to recover the scattering, the diffraction pattern, is what we call it. And then if you're able to recover many diffraction patterns from that crystal at different orientations, you would try to combine that information to try to determine something about the atomic structure of a particular molecule, which is shown here on the right. Now I'm going to briefly show an overview video that demonstrates this process from start to finish. So how do you come about a sample? Well, a sample can be in solution. It can be tiny crystals found in a solution, or it can be crystals found in powder. In any case, it's any situation where you might find an ordered array of molecules at the nanoscale or microscale. When these are present in solution, you would put them on a 3 millimeter wafer. You could either freeze them or dry them, or simply deposit them onto this wafer, then put them using this holder into the TEM. The TEM would then illuminate them with an electron beam as you're rotating to capture diffraction at various views or angles from that crystal. Those diffraction patterns would be collected using a detector that's sensitive to high energy electrons. And that detector actually sits farther below the interaction region. This is just simulating what you might see. You might see a diffraction pattern that looks like this, where constructive and destructive interference of electrons with the lattice in your crystal has been measured. And you can see all of these points of particular constructive interference are measured and assembled into this three-dimensional sphere of information. This three-dimensional information is then fed into reconstruction programs that have algorithms to try to invert this information and convert it into electrostatic potential, some continuous density that then represents your molecule. That continuous density is then fit using humans or algorithms to an atomic model. And so you fit an atomic model into this density and interpret the molecule that you are observing. Now, this can happen relatively quickly if your samples are ideal for this purpose. Now, if you have a very small crystal of a sample, say a small molecule, it's very well ordered and diffracts very well. It's robust, doesn't get quickly destroyed by the electron beam, and it's happy to be dry in atmosphere. Then you can put it into the microscope, collect a diffraction movie or a diffraction pattern, and then try to generate a three-dimensional structure from it. And you can see in that three-dimensional structure, you can identify the position of atoms by looking at locations where you see spheres of density. So you see, okay, this is where an atom must be. Now, that's in an ideal case. In practice, samples may be a little bit trickier to work with. And so a good percentage of the time, maybe 50% of your time in an experiment might be spent trying to figure out how to best put your samples onto an EM grid or into an electron microscope. And that can take minutes. Sometimes it can take longer. Then once you've put that sample into the electron microscope, not everything on that EM grid might be your crystal of interest or crystals of your molecule. There could be other things on that grid. And so some time is spent surveying, trying to figure out, okay, is this something that I'm interested in on the nanoscale? And you can see, for example, on this particular grid, there are a variety of specimens, and you would have to diffract from various ones to try to understand which ones you're actually interested in collecting data from. And this could take minutes, hours, or longer. And so once you've identified a sample of interest, you would diffract from it using the electron microscope. This would take some time and maybe minutes to collect a movie, but you might not be satisfied with a single movie from a single individual sample on your grid. So you might have to collect from various samples, collect a movie from each, and spend more time surveying. And so that could escalate the amount of time required for data collection and processing. So you'd convert those movies into some kind of format that is understandable by conventional crystallographic processing software packages. And we use a variety of software packages to do this, but our favorite is one called XDS. So XDS would take in these images. It would identify the type of diffraction that you have as crystalline diffraction, and then determine lattice parameters that gave rise to that diffraction, given the geometry of the experiment in the electron microscope. Ultimately, it would give you a set of intensities that, when transformed by a phasing program or a structure solution program, would actually give you density, a representation of your molecule in real space that you could then model using a modeling program and place atoms into. So that's the overall strategy. An example of that is shown here. So here are needle crystals of a particular, this is a peptide that grew tiny crystals. We couldn't really see these with the light microscope. It just looked like a bundle of needles in there. When we deposited them onto an EM grid, you can see relatively clear needle crystal here. You can also see there are other things on the grid. So there's some seemingly amorphous material here. There are also some fibrillar structures, sometimes even surrounding the needle crystals. And so we tried to identify a region on the scale of about a micron to illuminate with the electron beam and selectively collect diffraction from that region. And so you can see actually on this carbon film, there are many tiny holes. Each of those holes is about a micron. And so that means that the thickness of this needle is only a few hundred nanometers. The entire illuminated area is shown here in this sort of slightly darker gray color. That was the area that was illuminated by the beam. And then we placed an aperture inside the microscope to select diffraction only from the tiny tip of this needle. And so from that, we collected a movie that looked like this as we were rotating the sample within the electron beam. And you can see, again, spots that represent constructive interference at particular locations in reciprocal space. We can see that the extent of these spots represents a resolution of better than 1 angstrom for this diffraction. So we were really excited by that. This led to solving the structure of this molecule from this data set. Now, what would ordinarily that look like? So if you combined one or more data sets and put them into a structure solution program, something like a very popular program, ShellX, you would actually be allowed to guess essentially the structure of your molecule automatically using the program's algorithms. And so the program would take in those intensities that you saw, the constructive interference from those movies, and it would try to identify an assembly of atoms that was most consistent with that data. And that assembly of atoms, by its own metrics, would be judged for correctness based on its matching the crystallographic data. And so in this case, you can see an example of such an assembly of atoms that was identified by the ShellX program. And for anyone who's looked at peptides before, this very much looks like a canonical peptide. It's actually a hexapeptide structure. You can place atoms at these locations and then optimize or refine those coordinates to obtain a more or less final structure. And out of that final structure, about almost all of the atoms, 54 out of 59 atoms, were identified by the ab initio phasing program. The remaining atoms were identified during refinement as locations in space where density was present, but an atom wasn't placed by the program. And so this was done manually. This, of course, is even more obvious now as a peptide. In fact, you can read off the sequence, GNN, QQ, and Y is the particular sequence for this peptide, so you can see the tyrosine ring and so on. What you can also see, because of the resolution for the structure, are regions where hydrogens were not included in the model, but density is present corresponding to the locations of hydrogens in that model. And so it's nice to be able to actually place hydrogens in particular locations with confidence based on diffraction data. Now, using this approach, many labs have looked at a variety of materials, molecules, et cetera, ranging from zeolites to small molecules to proteins to peptides, and their structures have been determined. What inspired us to bring this technology to BioPACIFIC is its great application to small molecules and now increasingly to more and more complicated small molecules. Of course, this has garnered a lot of interest, both in the press and in publications. And what we've learned is that the structures that are determined by this method are relatively accurate, or at least we believe as accurate as the types of structures you would determine from much larger crystals using canonical X-ray diffraction methods. And so we're really excited that you can take canonical X-ray diffraction methods that work best with larger crystals and apply them to tiny, tiny crystals. These are crystals on the nanoscale, and get structures that are almost as accurate. Now, this was sort of borne out by collaboration with Professor Jose Nelson, who is also involved in BioPACIFIC and has led a good portion of his group in the application of electron diffraction techniques to complex small molecules. This was one of the early works that we collaborated on together, trying to determine structures of organometallic small molecules and crystals of those were diffracted on electron microscopes here on campus, we were able to determine a number of structures and showed that even at room temperature, we could obtain high quality diffraction from some of these molecules. And that was quite impressive. Now, that has made electron diffraction an analytical tool for discovery as well. There are a number of new structures that have been determined by this technology, and that number keeps growing. So now I think there are, over 100 structures that have been determined. Many of these structures were originally determined just to advance the method or further the method, but now the tide is turning, and many of the new structures that are being determined are completely unknown. And so they're structures that have never been solved by any other method before, and they weren't determined to advance the method, but they were determined for discovery purposes, so to solve actual research problems. The resolutions range quite dramatically. Some of the larger molecules, like these complex protein machines, might only be resolvable to 2 to 3 angstrom resolution, but some of the small molecules are resolved to better than an angstrom, and they're highly accurate structures. Now, this is not going on just at UCLA. There's a worldwide effort to try to improve the technology and apply it to a variety of substrates. This is just a smattering of some of the labs around the country and around the world that are developing electron diffraction techniques. And so we're really lucky to not only be at the forefront, but be able to participate in the advancement of the field and the application of that technology to really interesting research problems here at UCLA. And I've sort of labeled some of the substrates that are being interrogated around the world using this technique, and some of the prominent industry partners that are really interested in applying the technique to important problems. And so this is growing not only on the academic front, but also on the industry side. And so that's really exciting. So that was sort of an overview of the field and what you expect walking into the area of electron diffraction to try to understand structure determination from very, very small crystals. Now I'm going to focus on innovations in electron diffraction, also called microcrystal electron diffraction, or micro-ED, and what that means specifically for our community in BioPACIFIC. And so one of the things that we're really excited about is being able to tackle samples that might not be pristine, that might not form pristine crystals. And so, of course, if you're able to form large pristine crystals, you might just take your sample to the local X-ray diffractometer and collect a conventional X-ray diffraction data set. But if you have highly polycrystalline samples or crystals that are present in mixtures, heterogeneous mixtures of amorphous and crystalline phases, or so on, we're really interested in trying to develop methods to more routinely determine structures from those. And so some of the methods are sample preparation methods that we've adopted or developed. They include procedures as simple as simply exposing powder, lyophilized powder of your material onto a grid and then diffracting with the electron microscope, or drop casting and pulling vacuum before freezing, or treating suspension of molecules or a powder of molecules or whatever it may be, as a very sensitive system, and plunge freezing it using liquid ethane, much like folks in conventional cryo-EM setups do, and then transferring that in a frozen hydrated state into the microscope and collecting data from it in that state. Now, I want to highlight that this has been incredibly successful, and one of the most impressive efforts that I've seen recently is work by 2 BioPACIFIC groups, the Nelson Group and the Tun Group, and their effort is really impressive because it was not only trying to understand the structure of a molecule that was challenging to characterize structurally, but was just a tour de force, looking at very, very thin plate-like structures that would definitely not have been usable for conventional methods at all. And they were sort of a borderline case for electron diffraction. The lead student on this project, Lee Jun, Kim was able to collect movies that looked like this. And from these movies was able to determine a really impressive and complex structure with 6 different conformations of this fissurin molecule in the asymmetric unit. And that was really quite a tour de force. Now, another example is shown here. This is from a fellow colleague in the chemistry and biochemistry department at UCLA. And in this case, what was being investigated was peptide that formed polymeric structures. So there were these fibrillar polymers, basically. And the bonds here between the individual monomers were not covalent, but these polymers were quite robust. And each individual peptide included or contained a post-translational modification, in this case, a covalent modification that was altering its properties in some way. And so the structure of this polymeric molecule was really interesting to them. This was determined by electron diffraction also here at UCLA to atomic resolution, so they could see exactly where every atom in this core was. And this is sort of an unusual conformation for a peptide forming this type of polymer. It was this triangular structure, which would have, again, been very, very difficult to characterize otherwise. Now, in cases where there are samples that are even a challenge for conventional electron diffraction, we're really excited to try to push the limits of the technique at the moment. And rather than illuminating a micron size area or even larger, we're trying to change the illumination area to scale it down to the size of just a few repeat units within a crystal. And so we've partnered with folks at Berkeley Lab and are trying to set up the very same approaches here at UCLA to try to make pencil beams, or beams that are highly parallel but very small in diameter. In this case, you can see a profile of a beam that's only about 6 nanometers in full width at half max to try and illuminate very small regions of a crystal. And just to illustrate what that means, I'll show you a picture of a crystal here in the background and a conventional illuminated area using microelectron diffraction techniques in blue. And here you're illuminating a good portion of the crystal. You can see in this picture of this crystal that it's not homogeneous. In fact, there are structural variations that you can see sort of from not a very high resolution image of this crystal. You can see that there are structural variations here that are all being sampled by this blue circle. Now, in contrast, these small beams would be about the size of the red dot that you see on the screen. And the diffraction that we get from each is somewhat different. So this is the conventional diffraction. We're getting a diffraction pattern that we're getting in a selected area electron diffraction. And you can see that there are high resolution features in this diffraction pattern that extend out to about 1 angstrom in resolution. And by comparison, the diffraction is weaker from the small illuminated area, but we're still able to get regions of constructive and destructive interference. We can still identify a regular order to the diffraction patterns that is related to the lattice structure at that point in space. And so what we've done along with our partners at LBL is to try and raster scan that beam across a sample and either integrate the signal as you see on the screen here. So this is just integrated signal over relatively large area. As that beam is scanning, we're just summing up all of the information onto one single diffraction pattern. And this would be something equivalent to accumulating a single diffraction pattern using selected area diffraction. And you can see how long it takes to integrate here. This is about a second of information that is being recorded on this single image. Alternatively, we can look at individual diffraction patterns and not sum them up, but rather parse them out. And if you do this with a control sample, what you expect to see is an unchanging diffraction pattern, because if the sample is perfect, the lattice is perfect, no matter where you illuminate that lattice, you'll see the same diffraction pattern, the same type of interference between the lattice and the illuminated beam. And what we saw, even with the control sample in this case, which we thought was really pristine and regular, was quite a bit of variability when we did this experiment at Berkeley Lab. And so what we realized is that maybe our control samples weren't really behaving like control samples. And we were really interested at trying to understand, well, what type of variability can we see not only with control samples, but with real samples, and can we make something out of this? This variability is best understood if you look at an image of the crystal that we were interrogating. So this is a rod-shaped crystal. You can see the rod here sort of going diagonally across the viewing area. And you can see it's sitting on a piece of lacy carbon. The lacy carbon is down below and up above. And so it's sort of bridging across these segments of lacy carbon on a carbon wafer. And so as you're looking at segments of diffraction, so as the beam is raster scanning across this whole area, you can parse out diffraction from these various areas labeled 1 through 19. And what you can see is that diffraction over empty space just shows you the profile of the illuminating beam. So that's what the beam looks like that's impinging on the sample. If it impinges on a piece of the crystal, it immediately starts diffracting. And so you can see these higher order spots that appear as a result of the beam interacting with the lattice present in this region of the specimen. And so as you go farther and farther into the core of the crystal, you can see that that diffraction is changing. And we were trying to really not only parse out the differences in diffraction across these two-dimensional spatial maps of the beam raster scanning across the crystal, but also try to understand what the underlying physical basis might be for this behavior. And so we've developed algorithms to parse out those diffraction patterns. We can cluster diffraction patterns, even weak ones, to combine all of those that are similar into groups, and then those groups can get averaged to produce cluster averages that you see down here. This can be an iterative fashion. This can be done in an iterative fashion to try to optimize the grouping and identify the most coherent set of patterns that we can then group into relatively high signal to noise ratio patterns. So these patterns are not as noisy, and you can relatively clearly identify the type of lattice structure that is present and giving rise to these patterns. Because we knew what the structure would be, in this case, this was our control sample, we could also simulate diffraction from this particular type of lattice and then compare through template matching individual diffraction patterns from particular regions of space to the simulated diffraction patterns of a lattice at a particular orientation. And so when we did that, we noticed that this crystal was sitting with the lattice at a particular consensus orientation. We could identify that from the ensemble average across all patterns of that crystal. But there were regions of the scan that corresponded to subtle lattice reorientations. So this was a region of the crystal where the lattice was slightly tilted with respect to the consensus orientation. Now, the tilt wasn't large, but it was noticeable, and we could detect it from the diffraction patterns. And so when we put all of this information together, what we could see is that while quite a high number of the patterns across the scan represented the molecule sitting at a particular orientation in space, or the lattice being at a particular orientation in space, what we could sort of, in a hypothetical world, imagine there existing is ripples in that lattice that displaced the orientation of that lattice with respect to the consensus orientation. And this is what this picture tries to show. It just shows the molecule that presumably we would be sitting in this lattice at a particular orientation. And you can see that molecule being reoriented in space as a result of this rippling phenomenon. And so you can see the ripples here in space across the crystal. Of course, we are not visualizing ripples in individual molecules. This is just a representation of what's happening across that crystal. And so it makes us realize that really a lot is going on at the nanoscale here, and we're trying to capture that information as best we can with the best spatial detail that we can, while still maintaining atomic resolution information about the molecule's structure within a small region of space. And so we can do that for a variety of other crystals, and we find that there is this spatial variation even within pristine samples. We can map the spatial variation, and we can either group all of the information together across different spatial domains, or we can parse it out. Now, this is particularly helpful. The parsing out part is particularly helpful in cases where we want to determine an atomic structure of the molecule from series of domains or regions of a crystal that all represent one particular consensus state of a molecule, especially if the crystal is maybe polycrystalline, or if there are multiple crystals together and we want to parse out information from those different crystals. And so that's exactly what we did. We scanned a beam across another rod-shaped crystal. So you can see the scan area here represents about 3 microns in the vertical dimension and 1 micron in the horizontal dimension. And this particular crystal is about 300 nanometers thick. And so we could scan that beam across that area. We could then parse out regions of our scans that we believe represented the core of that crystal, the part of the crystal that was diffracting best. And this was done ex post facto. So after we've recorded all of this data, we've tilted that crystal to various orientations in a continuous fashion. So we start at one tilt angle and we're rotating that crystal in the beam and scanning. And then synthesize or generate diffraction patterns that would ordinarily have been obtained using a selected area aperture, but now are obtained using an algorithm that detects where the best diffraction is in space from these scans. And we get what would be equivalent to diffraction patterns from this crystal at minus 40 degrees, minus 20 degrees, 0 degrees, 30 degrees, and so on. And at the same time, we can actually reconstruct a real space rendering of that crystal itself. And so this is what we're actually reconstructing the crystal structure of, is this region of space that is segmented here. And so from that region, we're able to obtain an atomic structure for, in this case, a peptide molecule. And that peptide molecule is shown here. The density retrieved from the data that was collected is shown in the blue mesh. And then the ball and stick model is built into that density with reasonably good statistics. And so we're really encouraged by the prospect of doing this, not only for single crystals, but across many crystals. And others in the field have demonstrated that you can do this even across entire fields. So you can combine information from many crystals all into one single data set and then determine structures of molecules. They've even been provocative and suggested that you can not only determine the overall structure of a molecule, but also its handedness or chirality. And so we're really interested to apply those methods and find interesting research problems that they benefit. And so overall, we're looking forward to not having this background of large amounts of time spent sort of hunting for crystals and whatnot, but having adaptive serial data collection where we can maybe map out different crystalline phases or different types of specimens on the same sample, automatically detect the regions of space that are producing high quality diffraction, classifying those and processing all of the ones that are cogent, all of the ones that match the same type of structure, and producing an automatic structure solution, possibly using some new machine learning methods. And so this brings us, of course, to perhaps an elephant in the room, and that is that machine learning is getting so good at predicting structures that recently there have been really incredible breakthroughs in our ability to predict the structures of large molecules, in this case, proteins. And this has been most probably prominently demonstrated by the team at DeepMind that released AlphaFold 2, a new structure prediction pipeline. And almost immediately after the release of AlphaFold 2, many in the community started simply posting that's already benefiting their crystallographic efforts by providing them with starting models that yield structure solutions. And this list goes on, but here are some examples just from tweets that people have posted online. And the community has really embraced the predictions that have been generated by AlphaFold 2, and it really validates the approach of generating computed models to try to use as preliminary solutions or partial solutions for crystallographic problems. And so in our group, we've already started doing that, and we're excited to incorporate newer models for structure prediction into pipelines that will ultimately serve as a hybrid space where you can validate a predicted model by comparing it to the measured crystallographic data acquired by electron diffraction, and then sort of shortcut your way to a solution where the data may not be ideal and may not give you a structure ab initio, but could corroborate the predicted structure pretty faithfully. And so we've done this with large proteins like proteinase K. We've started out with piecemeal fragments from a model in the protein data bank. We've created an ensemble of those fragments that really fits electron diffraction data quite well, and then piece together those fragments into one coherent final model that gives fairly good stats. And so if this was in an unknown protein, for example, you might be able to do the same if you had distant homologs in the protein data bank, or if you could get a partial or fairly complete model from something like AlphaFold 2, we could generate presumably a final model from that if the fragments or the entire fold was close enough to be placed. And so all of that is really exciting, in particular because we at BioPACIFIC are excitedly waiting for a new instrument to arrive and be installed at CNSI that will be able to do all of the things that I've talked about today. This is a new Spectra C 300 kV instrument. The installation and commissioning will be again at UCLA CNSI Institute. And this will happen hopefully between September and November of this year. And it will be ideally operational and ready to use later this year. And we'll be able to, it will look like this box, but in reality, inside we'll really have a bunch of amazing TEM components, including some really high sensitivity detectors that will allow us to do pretty high resolution electron diffraction and yield high quality data to interrogate. In fact, I was at the sort of reveal of this instrument at the microscopy and microanalysis meeting before the pandemic, and I got to see some of its impressive results, including this very nice STEM image of a beam sensitive material. And so they were sort of demoing it live. And this is an example of one of the substrates that they were able to visualize with this beautiful lattice structure. And so I'm looking forward to having this instrument here at UCLA and being able to partner with everyone at BioPACIFIC to try to further our science using this technology. Of course, there are many, many folks that I have to thank for being involved in this group, in this work, including members of my group. I highlighted Dr. Marcus Gallagher-Jones, but others in the group that were involved in the work include Kelly Glynn, Ted Z, Maria Flores, Logan Richards, Umber Sa, and many, many others that have facilitated the electron diffraction efforts. The UCLA DOE has been also a strong support of electron diffraction, strong support system, including, and in particular, Dr. Duilio Casio, who is also advising the installation of the new Spectra C instrument. There are, of course, folks at the UCLA EICN and a number of micro-AD collaborators at UCLA and beyond, including Dr. Jose Nelson, whose group is involved in BioPACIFIC as well, and our collaborators at LBL. The National Center for Electron Microscopy has pioneered some of the nanobeam efforts that I've described today, in particular the scanning diffraction techniques, otherwise known as 4D STEM, and our collaborators at the Pacific Northwest National Lab. We wouldn't be able to do this without a variety of generous funders, and those are shown on the right with icons. And with that, again, I would just want to thank my group and everyone involved in the collaborations that I've highlighted today. And I'm happy to take any questions you might have.
Great, thank you so much. Jose, that was fantastic. A really nice blend of teaching us and also exciting science. So thank you very much. We do have a couple questions if you are ready to answer some questions.
Absolutely.
Okay, so the first one is from Matt Wallen, and he asks, could you mention the benefits of using electron diffraction over XRD?
Absolutely. So in the limit that your crystals are very small, being able to acquire enough signal from a canonical X-ray beam. And I mentioned some of the machines that are being built to try to extract signal from these very small crystals. And so one of the benefits might be if you don't have an X-ray free electron laser in your backyard or at your institution, but you have an electron microscope, maybe you can try using that electron microscope instead of the X-ray free electron laser. If you happen to be a hobbyist and you built an electron, diffractometer or something like that, it would be very much equivalent to the data that's generated at one of these X-ray free electron lasers. There are subtle differences in the sort of technicality of the approach and so on. But in terms of interaction, the types of data you would get would be somewhat equivalent. You could solve a structure from either one. Mm-hmm. Now, where theoretically there's a big difference is in the type of scattering you are witnessing. So in X-rays, you are witnessing scattering of light with matter. And photons, of course, don't have a charge and don't have mass. And so that fundamentally is going to differ from the type of scattering you're witnessing when electrons impinge on matter. Electrons, of course, have mass and charge. And so that charge is sensitive to electromagnetic potential or electrostatic potential in your molecule's crystalline array. And so in principle, you could see differences due to charge. You could also see interaction with light atoms because you're scattering from a screened Coulomb potential, which is derived from the nucleus itself and not from the electron cloud. And so that's what allows us, for example, to see hydrogen atoms more clearly than we would be able to see with X-rays. So I hope that there's a long list of things that I could go into, but I hope that I've sort of summarized the two key points, one being size and the other being the type of interaction that's happening that allows us to see slight differences between lighter atoms.
Mm-hmm. That's very helpful. Thank you. And then we have a question from Matthew. What limits the periodicity you can detect in your diffraction patterns? Is it Debye Waller effects, electron dose, or something else?
That's a great question. The signal that we're seeing is coming from a periodic structure. And so if your structure is not periodic at all, let's say you have a purely amorphous structure, like a glass or something like this, we would not be seeing the type of diffraction that we see here. We would not see these key locations of constructive interference that would allow us to then determine a structure for that assembly. Now, why would a structure not be ordered on this scale? I think the answer to that is also very complex, and I could name a number of reasons, but one of them that you brought up in particular is an effect that's due to damage of the lattice by some means. Now, that can be because we are observing it. So some molecules, when you illuminate them with the electron beam, they're going to respond to that. And so there's going to be some kind of modification that's going to happen. That is commonly, of course, referred to as radiation damage. So you're irradiating the molecules, there's going to be some sort of damage. We try to limit that to the extent that we can, but of course, it's always present. So it can be that you're irradiating them, and that's why you're not observing crystallinity, because you're destroying the lattice as you're irradiating it and causing, runaway chain reactions or something. It could be that you're damaging an existing lattice as you deposit things onto a grid. Maybe the vacuum is pulling on that lattice and collapsing it in some way, because that lattice depends on ordered gases or solvents or something to maintain its integrity. And so there are a variety of reasons why a pre-existing lattice might collapse. But if you don't have a lattice in the first place, then we're sort of back to square one.
Mm-hmm. Great. Thank you so much. I have a question quickly.
Yeah.
I know we only have one more minute, but
yes.
You mentioned how you can just throw powders, sort of lyophilized powders on the surface. So how does it work? When do you know that you're going to get structures from that? Or is it always, or?
That's a good question. I wish I knew. If I knew, we'd be in a better place. No, I think in reality, there are a variety of ways to pre-screen samples that will give you some corroborating information. Sometimes you can use a light microscope or some other type of analytical technique to try to determine if you have things that might look crystalline because they have, features that appear geometrically crystalline. And so that's one way. Another way is because maybe you have evidence from powder X-ray diffraction or some other corroborating technique that indicates that you have periodicity present in the sample. In those cases, you can have a high degree of confidence, but it doesn't rule out the possibility that you have a small subset of your sample that does have the particular type of properties that you like. Maybe they're ordered, but it's a small, overall percent of the mass of your powder or something like that. And so the best way to figure that out is to just deposit some on the grid and screen. And that's where time surveying is spent, is trying to identify those crystals that do diffract.
Okay.
Yeah.
Thank you so much. This is very helpful. I really appreciate you coming, and we learned a lot. I know I learned a lot, so thank you.
Of course, thanks.